Welcome back to the OIS podcast. In this episode, Dr. Arthur Suckow, a self-described drug hunter, discusses his pathway to becoming CEO of DTx Pharma, a company working on overcoming challenges with drug delivery. Let's listen.
Welcome back, everyone, to the OIS Retina podcast. This is again Firas Rahhal of Retina Vitreous Associates here in Los Angeles and also of Excite Ventures in New York. And I have today as my guest a good friend of mine and a colleague, Dr. Artie Suckow, who is the CEO of a company known as DTx Pharma. We're going to hear a lot about that today. Artie has a long history in biomedical sciences. He has a PhD from UC San Diego in biomedical sciences and pharmacology. He's an expert in this area. Obviously, we can't wait to hear about some of the science that they're doing. And he, I think, is the co-founder or the founder of the company. Artie, first of all, welcome. Thank you for coming on the show with me.
Well, thank you for having me, Firas.
Tell us about your own personal background. I read from some of the aspects of your bio that you had previous positions with AstraZeneca, Johnson & Johnson, both very famous right now. They're in the news a lot, as you know, for maybe different reasons, although you can share with us if there's any overlap. And Regulus, something I'm not familiar with, Regulus Therapeutics. And again, you had previously received a doctoral degree from UCSD in biomedical sciences pharmacology. Tell us a little bit about some of those previous positions, and were you working at that time with the fatty acid molecules and the peptides conjugated to fatty acids, as I read about?
Yeah, so maybe I'll start. Well, it's interesting how things come full circle. So I did my undergrad at University of Delaware, and I actually did ophthalmic research at University of Delaware. To be honest with you, I thought that was the last time I would do research in the eye. I studied the cornea. But anyway, come to, how I came to know fatty acids, did my PhD at UCSD, all in diabetes. I studied pancreatic islets, the insulin secreting cells, went to Johnson & Johnson, stayed in the diabetes space. From there, like you said, AstraZeneca, again, diabetes space. The difference between J&J and AstraZeneca was J&J was small molecules. AstraZeneca was biologics like antibodies, peptide conjugates. And then, Miss San Diego, the beautiful weather in San Diego, and joined a company called Regulus Therapeutics. And that's where I came to know the problem that DTx works on, delivery of RNA therapeutics. But it was really, work that I had been involved in at J&J in working on fatty acid receptors for small molecule diabetes drugs, and work at AstraZeneca looking at fatty acids as a means to protract the half-life of peptide diabetes drugs that sort of served as the inspiration for DTx.
So the work at Regulus was with RNA therapies, and that did involve these long chain fatty acids at the time too. What were you doing there exactly?
Yeah, so no fatty acid work really at Regulus. So Regulus was, there's all sorts of flavors of RNA therapeutics, and they worked on microRNA, which is kind of nature's siRNA. And it regulates many different processes, but it was there. I was in charge of sort of thinking about new ways to deliver these medicines. It turned out that they underwent a lot of change, and throughout my year there, I worked on CNS, eye, diabetes, and NASH, as well as delivery. So that's probably more a reflection of the change going on there. But what I came to know there was the delivery challenge with RNA therapeutics that has sort of plagued the space for, over 30 years, right? Cells don't take these molecules up at all, and they're rapidly cleared by the kidney. And so, outside of like local indications like the eye, being rapidly cleared by the kidney precludes exposure to like heart, muscle, where there's lots of opportunities for RNA medicines, but if you can't get them there, how are they going to work?
So when you were at Regulus, you were working in the RNA therapy space, but they weren't specifically like you are now working on a niche area of delivery of the molecules. That's something you observed in the challenges in doing the work itself.
Yeah.
Is that how the genesis of DTx came about? Tell me about how these positions ended up leading you to where you are now and how it led you to form DTx.
Yeah, there's lots to this story. Part of it is, why did I even want to start a company to begin with? To part of it, how did the genesis?
Because you're masochistic.
I do go back and think about what I was thinking 3 years ago that kept me going from day to day, from time to time. But anyways, the genesis of DTx is kind of, twofold. And maybe I'll start with why was I inspired to start a company, and sort of my career path and what I was thinking as I was at AstraZeneca and J&J, and then how DTx came. And so it turned out while I was in grad school, there's something called the minor proposition, and you have to come out with your own idea outside of whatever your thesis work is. And so I presented that idea to my committee, and shortly thereafter, one of the advisors had said there was an investment group that was interested in investing in this idea, unrelated to DTx. But it ended up getting funded and became a lab that I worked in that was outside of the university, that I reduced it to practice. And so what I realized pretty quickly there, even though the idea worked, was that having 2 years of experience at grad school was not sufficient to build a biotech company. But so I stored it in the back of my mind that, it sort of became my ambition to get appropriate experience in drug development, so one day I could come back and do that. And so finished grad school, but while I was at J&J and AstraZeneca, I was always trying to interact with people far smarter than me who had more experience, to learn all about different aspects of drug development. It was kind of why I moved from large pharma to biotech. Was at Regulus, I maybe aspirationally thought that I'd learn how to build a biotech, do relationships, raise money, etc. Obviously, that didn't work out. But fortunately, I guess for the prospects of building a business, what I did learn at Regulus was, as I mentioned, this delivery challenge, and why I thought, with my colleagues Adam and John, thought that fatty acids were the solution for the delivery challenge, was essentially because they kill two birds with one stone. And so, every cell in your body has a mechanism to take up fatty acids. And so you can imagine leveraging those mechanisms to bring these molecules into cells. The other reason we really liked fatty acids is they were a tried and true mechanism. And I learned this at AstraZeneca, for promoting biodistribution. So there's 4 multi-billion-dollar peptide drugs in the diabetes space that leverage fatty acids on the peptide for half-life extension. And so what those peptides do, or what do those fatty acids do, is they bind to a protein in the blood called albumin, and albumin kind of serves as an Uber to maintain exposure of those drugs over prolonged periods of time. And so that was the same fundamental problem that RNA therapy, peptide therapeutics, what fatty acids did for peptide therapeutics is, the same problem that RNA therapeutics face, rapid clearance by the kidney. And so, saw that as really underexplored relative to what others had done with fatty acids in the RNA therapeutic space. And that was sort of the inspiration for the idea, gained, traction with investors, at least early on, to reduce it to practice.
That's amazing. So the fatty acid moieties have been, I won't say commonly, but previously used to attach to peptides to increase half-life, say, in the circulation. Does that also relate to their penetrance into cells like you're talking about here? Or is that strictly because of the albumin phenomenon that you discussed?
Yeah, in that context, it's all about, the drugs don't need to get into cells per se. Okay. You're trying to get them to cells to interact with, like the insulin receptor, or there's another drug called GLP-1 that's important for promoting insulin secretion in beta cells. So in that context, those success stories are really about keeping drugs in circulation longer.
Did you have exposure to this fatty acid concept while you were a grad student? Is that when you first thought about it? Was it later on after you saw the challenge in RNA technologies entering cells that it came to you that this might be useful there?
I never thought that I would use fatty acids again. When I left J&J, we worked on these fatty acid receptors for diabetes. The fatty acids were ligands for these receptors. Wrote a paper and a review article, thought that was the end of my career in fatty acids. Came to, AstraZeneca, where we used fatty acids on peptides for half-life extension. Again, thought that was the end of fatty acids. And then, when I saw that the setback in RNA therapeutics was delivery, I sort of reminisced on my experiences at J&J and AstraZeneca and say, man, it just seems like there's probably a way you can, I didn't necessarily know the exact conformation or configuration, but it seemed like, as a reasonable hypothesis as any, that this could work. And in grad school never even thought about drug development in any meaningful way.
You're too humble to say it, but it seems to me as a simple guy, that's how great scientists come up with great science. That story is a great story of how one puts together two pieces of a puzzle that are remote from one another in time and space. And this is how great ideas come about. You deserve a lot of credit for that. Would you say this is novel to attach these moieties to RNA technology? Is this the first DTx and you the first of its kind?
I won't claim that we are the first company to attach fatty acids to siRNAs or antisense oligos. People have tried it since 2006. What I think is, in some ways, there's a fortunate coincidence of cross-fertilization here. Having seen this in other contexts, that it seemed obvious to me that folks had tried to attach fatty acids to siRNAs and tried all different ones, but sort of didn't have the same appreciation that we brought to the space of how they actually interact with receptors and albumin to be able to leverage that knowledge. So, people tried, everyone's probably heard of cholesterol, right? People tried attaching cholesterol, and it worked reasonably well, but cholesterol hasn't been safely deployed for anything, whether peptide or otherwise. So that didn't work out. And you'll even see, publications all the way through last year of people attaching a single fatty acid. So the idea of using fatty acids is by no means novel. I think what we, brought to the table was we had conviction that fatty acids were the solution, and we more aggressively pursued, different numbers, orientations, lengths, all different kinds of variables. The way I kind of think about it, honestly, is like small molecule drug development, right? You get a hit, it's like a 1 micromolar hit. It barely binds, it's active. And so that's kind of where the field was, when we came to the scene. And then we did SAR, more chemistry, to optimize the way those are presented to cells and to make, orders of magnitude improvement. So I guess that's the way that the incremental benefit that we brought to the space.
I understand. So that brings me right to the topic I want to move into, which is the company, of course, that you founded. It's called DTx. And because I've never known this, even though for the audience, I'm an investor through Excite. Our group is one of the investors in your company. How did you come up with the name DTx? What does it mean?
Yeah, this is kind of, you're going to get a kick out of this. So
I've been dying to ask you, actually. This gave me an excuse to ask you.
So there's multiple answers to this. So I think the honest answer is the first 100 names we came up with, maybe more, because I remember the email conversations between the founders, were all, either taken from a website perspective, or we couldn't afford the domain. So I guess we ultimately settled for delivery technologies. So that's what the DT stands for. And then where you're going to get a kick out of this is the X actually comes from, when I was younger and I had instant messenger, I used to use the X as spaces between initials in my screen name. And so my screen name was Michael Jordan, MJX, SPXDR, Scottie Pippen, Dennis Rodman, and the X's were spaces. So DT was taken, so we added the X as the space. What was cool about the X is on the logo, almost immediately you could imagine building like a little siRNA off of the X. So you got, be careful what you ask for in the answer to the question.
That's a great answer. It's very honest. It could have easily been DT underscore given that.
I guess so, given.
I like DTx. By the way, X's and Z's are very popular with the Madison Avenue drug name developers, the guys who, because I did this as a resident for extra money when I was a resident in New York City back in the very early 1990s, they'd have focus groups and they would want to name drugs. Many of them are out on the market now. And sure enough, 20 years later, I saw these glaucoma drugs coming out that had Z's and X's in them because these Madison Avenue gurus knew that would be catchy. And it really, if you take a look at an inventory of ophthalmology eye drops in the commercial world, a lot of them have X's and Z's. So you did well the Madison Avenue guys would have agreed with you. So look, let's talk a little bit about the technology. It's fascinating. At the same time, it's complicated. It's hard for some of us regular folk to understand it. It's siRNA, and for everyone listening, small interfering RNAs. Tell us a little bit broadly and as simplistically as you feel you can deliver the message. What does this mean? How does the siRNA work? What is the therapeutic effect that might be gained by using this technology?
Yeah, so my simplistic way of, explaining this is, DNA, you've got the code, the DNA. That DNA, is, it's almost like a cookbook. It's transcribed into a message, and then the message results in a protein that does all the work. And so the way RNA drugs work, or siRNAs work, is they essentially chop up the message. Right? They cleave it, they clip it, they make sure that there's not going to be any message around. And then therefore that prevents the expression of the protein. And so why do you want to get rid of the protein? And so there's lots of, we hear about mutations and genetics. And so there's mutations in genes that lead to proteins that are toxic to cells or take on functions that you don't want. And so by, intercepting the message, or preventing the expression of the message, you can prevent the expression of the disease-causing protein. And maybe it's helpful because this is an podcast. I could use an example gene like rhodopsin, right? We know in retinitis pigmentosa, for example, mutations in rhodopsin lead to blindness. And so in this case, you'd use an siRNA therapeutic to get rid of the mutant rhodopsin to prevent the disease. So, or prevent retinitis pigmentosa. So, hopefully I broke it down simple enough.
No, it makes a lot of sense the way you propose it, which is ultimately the protein is the product, and along the way you're breaking up the messaging system, the building system. So this is going to block the formation of proteins. You would never use it in reverse to try to create a new protein that's better. That's a different form of gene therapy.
Right. Yeah, exactly. So that's, so broadly speaking, RNA therapeutics, it also encompasses what you're suggesting, kind of replacing the mRNA. And it's timely because, that's the mRNA technology is what, Moderna and Pfizer are using for the COVID-19 vaccines.
Excellent. That's very clear. and for some of my listeners who've been around for a while, treating ophthalmology patients since the 90s, I'll bring up a product that maybe you can comment on. This, as you mentioned, this has been a concept sort of circulating biomedical space for 30 years. In ophthalmology, we did have some brief experience with it in the 90s. I believe it was an siRNA, but it may have been something called Antisense. And if there's a difference, maybe you can help us what those differences are. The product was called later, I think commercially, Fulmiversin. It was used for CMV retinitis. I knew the person who developed it, or helped to develop it in New York, a great clinician at Mount Sinai. And I was treating a lot of CMV retinitis in the 90s, and this was fairly popular as it came on, but it didn't last too long. It may have been because CMV as a disease kind of diminished away with much better HIV treatment. Do you know about that drug, and what was that exactly, and what's the difference, if any, between siRNA and Antisense?
Yes. So maybe I'll start with the latter part of the question, which is there are, as you kind of elucidated, there are different flavors of, gene suppressing RNA therapeutics. And so there's siRNA that I just described. And then there's, if we get into the, hardcore molecular biology, the difference between siRNAs, they work in the cytoplasm. The antisense molecules, they work within the nucleus.
Okay.
And so that's a fundamental difference. They just use different, protein complexes to accomplish the same end, to chop up, to get rid of the message. And so maybe coming back to Vitravine, I think you actually answered the question on what happened to that drug. It went extinct because of heart therapy.
Yeah.
I think it was, I'm blanking on the name. Who was it? In any case, whoever it was withdrew marketing authorization in 2004 and 2006 because there was no market to sell the drugs to anymore. I guess probably other points related to siRNAs versus antisense. Our enthusiasm for siRNAs, maybe I'll start there, is they're quite a bit more potent, anywhere from 100 to 1,000-fold more potent than an ASO to accomplish the same end, repression of gene expression. They also have a really long duration of action following a single injection. They can last as much as a year, which is obviously really attractive when we talk about the eye. And then one other, maybe this is going into the weeds a bit, but I think your audience will be very familiar with some of these drugs. Is antisense, not only can be used to repress the expression of genes, but can it also do something called exon skipping? And you'll have heard of many of these drugs. I'm sure you've heard of ProQR. Mm-hmm. They've got a product for retinitis pigmentosa, for LCA. And the reality is, those are exon skipping is where antisense has shined. And so I'll actually defer to you on whether you want me to take a stab at explaining exon skipping or whether we want to move to another topic, but just, there's lots of flavors of these things. They can accomplish different things for different purposes.
I actually would like to hear, but I'm going to let it go because I know you could do it well and everyone would understand it, but I want to get into some of the clinical stuff that you and I have, of course, talked about and is more familiar to me and most of the audience members. By the way, I think that company you were looking for, I think it was called ISIS Pharmaceuticals that brought that product to market. And it was a good product. There were a lot of good products for CMV in those days. So the first eye indication for your product in the eye space, again, is retinitis pigmentosa, if I'm correct. How did you come to this? And specifically within that disease, what are you aiming at? What's the product going to do?
Yeah, so we were always very interested in the eye for this class of medicines. In some ways, we viewed the eye as a good place to understand the technology without the confounding effects of, biodistribution or rapid clearance by the kidney. We also, really like the eye because it's a compartmentalized approach. This class has a really long duration of action following a single injection. And so I'm sure you're familiar with all the VEGF products and trying to take them from monthly to quarterly to 6 months to yearly. And so we always were attracted to using RNA therapeutics because they have that built-in long duration of action. And then there was also, reasonably compelling advantages versus gene therapy in terms of, the number of cells with a genetic lesion that you can access, so distribution. So how we came to work on RP is interesting. We came across a literature through actually someone we both know, Dan Chow, that it's an approach that's meant to be agnostic to the underlying genetics driving the disease. And so, we put that up against other programs in RP that would more specifically address an underlying genetic lesion. And so, demonstrated it worked in, wild type mice, and then a couple different, I think 3 different rodent models of the disease, and then pitted up against other projects in the company and went out to the KOLs in inherited retinal degenerations and asked them, which of these potential products are you most excited about? And our approach to, our agnostic approach to the underlying genetics driving RP was unanimously selected. And so we've been pushing that forward ever since.
You're talking about the one that's specifically agnostic to the genetics of the RP.
Yeah, and I think one of the challenges, you probably know this as well as I do, one of the challenges with RP is it's genetically diverse. Yeah. There's 100 genes, 300 mutations, and it's really hard, to isolate any one of these genes and develop a drug for it, because there's not many patients with any specific lesion, and then they're not typically at a time point where intervention is likely to work. And so that's why I think everyone was attracted to this approach that could work in more than one mutation.
So that makes a lot of sense, and I agree with your KOLs, by the way, with the strategy. And we've talked about this, you and I. So it would be applicable to most all comers with RP. What about applicability to, say, different stages of disease? Do you think this product would be more likely to be helpful for early stage disease, mid, or even advanced stages at bare light perception, or those patients?
Yeah, I think it's more in the prophylactic category than sort of late stage. One of the major challenges with retinitis pigmentosa, at least as I understand it, not having studied it for my entire career, is that what happens is you have the death of the photoreceptor cells. And so as I understand it, if you don't have photoreceptor cells, none of the treatments are going to be able to work. And so just by kind of definition of the disease, you have to start early to prevent the death of those photoreceptor cells. And that's, so the earlier the better, I think, would be the strategy for this particular approach.
Yeah, I agree totally. I think for those really late stage disease categories, there's different paradigms, which we won't get into great detail on here. Optogenetics is being, attempted there. As you know, from Second Sight, there have been some electronic, devices attempted there. But for a product like yours, yeah, early and mid stage makes a lot more sense. Where are you with the preclinical development now of this product?
Yeah, so we've nominated, I guess, in drug development, what we call clinical candidates now. We did that earlier this year. And so that means that we're triggering the characterization and the safety that would support first in human dosing sometime in 2022. And so we're moving through monkey and rabbit studies that would support a safety package, that would allow us to advance the program.
So you your goal would be to have start human clinical trials in 2022 with this product?
Yep.
That's pretty near term. So you're already doing currently or are about to do non-human primates?
Yeah, so we're doing, we're at the dose range finding stage of our tox studies, and those are, usually you do those in two species, and it's safe to assume that one of those is non-human primates.
Okay, great. That's pretty far along. What about the corporate development? Again, for full transparency, I as a member of Excite, I'm an investor, so I'm aware of some of this. I'm aware that you had a great close to a raise recently. I think that's been public. What can you tell us about that recent raise that you feel comfortable to talk about, and where do you expect that to take you, and who is involved?
Yeah, the big announcement is we raised, about a month ago, we raised $100 million, from a combination of our existing early investors, as well as RA Capital, Access, Janet, Cormorant, Surveyor, and Eli Lilly, who was also an investor in our Series A. In terms of where we expect that to that capital to get us, specifically with respect to the eye, I mean, our goal is to get into human studies with that capital, demonstrate safety, and identify appropriate doses to move into studies where we would understand whether this is a potential mechanism to prevent the pathogenesis of RP. Of course, that's a lot of money, and we're doing other things. And so we're working in other therapeutic areas, including neuromuscular applications of the technology. The goal is to take one of those programs into phase 1 and potentially even some early proof of concept in patients there. And then, we have aspirations to, break open other therapeutic areas for this class of therapeutics. And, we want to remain the leaders in leveraging fatty acids. And so to overcome the delivery challenges in the space. So there's some effort to understand it in new areas like the CNS or oncology.
You got me to a point I wanted to get to with you, and thank you for that, the other programs you're looking at. And I knew that you were in the new neuromuscular space or pursuing that. I didn't know about the oncology. Have you already started any work in the oncology space now, or is that for later?
That I would just say for later for now. It's on the to-do list.
Yeah. What's ruminating inside your brain, I don't count that as now. We'll count that. That's the now, the real stuff later. I get it. I'm glad you mentioned Eli Lilly, because it's one of the things I wanted to bring up. I know they were an early investor with you. I know I had spoken with them in a group call when we were, doing diligence on DTx with you and with them, and we were very impressed with their level of knowledge and understanding of this space. We were very happy to join in the round when they came in. How's it been having a company like that, a strategic with such experience and broad wings, to come in so early? There's obviously value add. How did you view that?
Yeah, I mean, we've gotten lots of value out of that relationship. I mean, first and foremost, for the way this company was kind of built up, from the ground up, it was really important that we had external validation of what we were doing, outside of the local San Diego ecosystem. And so, by Eli Lilly taking a look at the technology and saying this is sufficiently interesting that we'll put some money behind it, was really important in our development. We also got the benefit of having their CSO at the time of RNA Therapeutics, Andrew Adams, on our board, who's understands space really well. He understands how to create value in the space, and it allowed us to get some insight into how potential partners, potential acquirers, look at these opportunities and the data sets that you need to have to create value in the area. So I mean, very significant inflection point and very valuable to have that relationship.
I know you've added some great people in recent months, and obviously you just had this excellent raise. Congratulations. Are there some corporate growth areas that you're eyeing right now, or are you kind of in a steady state and just going on to do more science?
We're definitely not in a steady state. I mean, we are a data-driven and R&D company, and so, a good chunk of our investment is in filling out our team with folks that are experts in ophthalmology, experts in neuromuscular diseases, both internally and externally. Now that we're bigger, there are we have to build out the operations arm of the company. So we will be looking to add a CFO and a VP of finance, clinical operations, et cetera. And maybe we've discussed this, but we brought on Denise Bevers as our chief operating officer, who has lots of experience building companies and taking companies public. Brian Lafitte, our CSO from Novartis, and Chuck to lead the chemistry, and he has 20 years of experience in oligo therapeutics. So those folks, much more knowledgeable about all those areas than me, are really leading the charge in terms of building out the talent that will keep this company remaining leaders in the space.
And that probably means you get to take off some of the hats, but it won't reduce your workload. I know you're a hard worker and you've led this company amazingly. Were you previously functioning as the CSO as well as the CEO, so to speak, prior to bringing in the recent folks?
As you know, I've been a drug hunter for my whole career, and so when
be careful how we reference that later, but
drug hunter by day and night. Anyway, I mean, in some ways, when we started DTx, John, Adam, and me, I didn't feel comfortable, calling myself the CEO, because my background was really CSO background, and so I guess more recently, as the company has grown, and I've had to prove to myself that I was capable of doing some of the non-science related aspects of a CEO. Thought, I guess I feel in my own head I transitioned to a CEO, but also I think it's really important. As you know, I spent a lot of time raising capital, meeting with investors, putting infrastructure in place to support the company. I thought it was important, especially with the programs, to bring on someone like Brian, kind of pass the baton, who has done this over and over again, more times than I've done it, to really take some of the or divide and conquer, I guess you could say, is the best way to put it. So was really really enthusiastic to bring him on board to help bring these programs forward.
Well, usually it is true that the scientist guy, which is you for sure, might have to give some thought to, can I be the CEO guy? Well, I'll say, as an investor and now your friend, you've done it in spades. You did train yourself in whatever way. And if this recent raise isn't proof, there's 100 million reasons that you have been able to successfully, and I mean this only half jokingly, to successfully build the infrastructure and lead the program, not just with science, but with management, that people are believing in you and they're putting their money in for a good reason. And you should be congratulated. That's a hard thing to do, to wear both of those hats, and you've done it very well. And you're probably pretty tired a lot of the times. One final question, really on the eye. We'd had some very casual conversations, you and I, in the past about indications outside of RP. You mentioned the non-eye stuff, the neuromuscular. What about in the eye? Other indications you're considering?
Yeah, I mean, we definitely are looking for other opportunities in the eye. I would say nothing has really gained traction in the way that RP has gained traction. And we've looked, as we've looked across a whole host of, potential opportunities, not limited to, the genetic variants of RP we discussed, dry eye, uveitis, Fuchs, wet AMD. And so, what has limited us or sort of prevented us from taking a shot in some of those areas is in part the competitive landscape. When we pick indications for DTx, I always say we pick indications where if we're successful, we will win, because we're addressing either the underlying genetic lesion and we can be the first to do that, or because the competitive landscape in other opportunities is favorable. But we're definitely still working through the process of identifying other opportunities in the eye, and I'm sure there's one there that fits the criteria. We just aren't ready to announce it just yet or commit to it just yet.
That dialogue to be continued. We'd love to have you on back later as things evolve for sure, and I mean that. Suckow, Dr. Artie Suckow, thank you for joining me and the rest of the team at OIS for our podcast. This has been fascinating. You're doing a great job. You're to be congratulated, and I appreciate you coming on.
Thanks, Firas. Thanks for having me, and appreciate all the thoughtful questions.
Thank you for listening to Dr. Suckow's interesting and inspiring discussion. Be sure to register for the upcoming OIS Myopia Innovation Showcase taking place on April 29th. And come back next week for another informative discussion here on the OIS Podcast.